hoping that Dr. Annie Pardo has found her voice here in the last 30 seconds. She's from Mexico and part of the dynamic team that's informed us so much about fibrosis.
This is for okay. Good morning. And I will show you some data related with MMP19. Since the first microarray studies in IPF, one of the things that we were aware was that many MMPs were upregulated in IPF lungs. That was in some way a surprise, especially for some type of enzymes like MMP1 that is a really interstitial collagenase. That means that its main substrate is fibrillar collagen. But in many studies after this, it has been consistently shown that many of these enzymes are upregulated. Here you can see a representation of all the family of MMPs and how are they regulated or how are they expressed in IPF lungs. It is important to remember that together with their function as molecules that are able to cleave extracellular matrix, and that's the main reason for which we know MMPs, when they are cleaving this extracellular matrix, they show many growth factors that are hidden in the extracellular matrix, and on the other hand, these enzymes are able also to process and activate several chemokines, cytokines, growth factors, etc. Now we also know that these enzymes not only have extracellular functions, but some intracellular functions are emerging with, in our knowledge. So in this work, we followed a different strategy. Not analyzing the whole lung, but separating different areas by laser caption of the IPF lungs, and we took in the same patient normal-looking epithelial cells in non-fibrotic regions, epithelial cells adjacent to fibrotic regions, and dense fibrotic regions. And we made microarray analysis of all these regions, and 6,308 we found differentially expressed genes. And in this way, it emerged a new MMP that hasn't been observed before. That's MMP19. MMP19, that there are few studies related with this relatively new MMP. First things we did was to see what substrates were reported for this enzyme. And as you can see here, that's not a true collagenase, meaning that it doesn't degrade fibrillar collagens, but it's related mainly with basement membrane molecules like type 4 collagen and some others, as you can see there. So first we validated the study. That means that we went to see if the protein was present, and here you can see that MMP19 is in the epithelial cells, as you can see them here, and also we found it in the endothelium. So next, we went to see if in the bleomycin model, the enzyme was also increased. No, sorry, this is data with the protein in humans. And here you can see that the protein is also increased in IPF lungs, and in BAL it is also increased. And here you can see in the experimental bleomycin model that you can find the enzyme also upregulated, and the same thing at the protein level. So what we decided to do next is to look for the MMP19 knockout mouse and to ask several questions. First of all, we analyzed the inflammatory response, and as you can see here, the MMP19 deficient mice develop less inflammation compared to wild type. And this less inflammation is mostly related with neutrophil presence, that there are less neutrophils in the MMP19 knockout mouse, and this correlated with myeloperoxidase activity. So at this moment, we were sure that what we were going to observe is that these mice would make less collagen. But we found exactly the opposite thing. MMP knockout mice developed a stronger fibrotic reaction. So here you can see hydroxyproline that is increased, and what we also observed is that we could see in this model true fibroblastic foci, as you can observe, and here stained for alpha smooth muscle actin. So it was not having MMP19 made these mice they made much more aggressive fibrotic response. And we went to look for some of the substrates that this enzyme is able to cleave or to degrade, and we looked for tenascin C, and what we saw here, is that, at least we interpreted it that way, that there was more accumulation of tenascin C in the knockout mice, meaning that this is not only a substrate for MMP19, there are many other enzymes that are able to cleave it. But here you can see tenascin C is accumulating. So, meaning that it's a substrate of MMP19. And now I'm going to show you several studies related with epithelial cells and MMP19. We analyzed what happened if we silence MMP19 in A549 cells. And as you can see here, this is silence. And we, after that, made a wound closure assay. And as you can see here, it's reduced when the MMP19 is knocked down. And on the other hand, if we transfect MMP19 to epithelial cells, what we observed is that there was increased cell migration. Then we decided to analyze what genes were upregulated together with MMP19 when we transfected A449 cells. And we found several genes that were upregulated, and we focused our attention on PTGS2, that is COX2, and I will show you some data of the relationship between these two molecules. Here you can see that when we transfect MMP19, then also COX2 is increased compared to the control. And when we silence MMP19, also COX2 goes down. And here you can see also that that was for the protein, the same situation. It was increased when we transfected and decreased when we silence. In mice lung fibroblasts, we can observe exactly the same thing. In MMP19 knockout mice, COX-2 is diminished compared with the wild-type animals, and the protein in the bulk fluid the same. So there is some kind of relationship between these two molecules that they go together. Actually, there is a nice study lately showing that COX-2 also is related with MMP19 in with MMP9, sorry, in pancreatic cells. So meaning that COX-2 has a bidirectional relationship with some MMPs. And interestingly, as you well know, also COX-2 knockout mice develop more or increased fibrotic response as in the same case of MMP19. Finally, we localized COX-2 in epithelial cells. We show that it is there and that it colocalizes with MMP19 in the lungs. So what does this mean, or we are trying to understand how comes that a molecule that seems to be increased seems to have a protective role. So we postulate that MMP19 is an antifibrotic mediator that has a close relationship with COX-2. We do not know, and we are trying to understand this, what are the mechanisms by which these two are related. And I haven't had time to show you our studies with fibroblasts that behave exactly the opposite to epithelial cells. That's meaning that fibroblasts that don't have MMP19, they proliferate more, they migrate more, the opposite to what we observe with epithelial cells. So here we are proposing that MMP19 related with COX-2 has a protective role. And as you well know, it has been shown that MMP7 has a pro-fibrotic role and actually is a marker of pulmonary fibrosis. And we have previously shown that it has also a close relationship with other molecule that is osteopontin. So what is this reflecting? We think that it's reflecting really what is happening in the fibrotic lungs. That is, there are many protecting molecules, there are many promoting molecules, and the lesson here is not only the thing that one molecule is upregulated means that it has a role in the pathogenesis of the disease. Probably it's a reaction of the cells to protect from the injury. So we are trying to understand more of this and following more studies. Thank you very much.
I think they could remember. 3 days.
Annie, hello. Very nice talk, Annie. Before I ask my question, I'd like to congratulate you on providing additional evidence to the already fairly sizable body of evidence that points to the notion that inflammation inhibits fibrosis rather than causing it. But my question is, what is known about activation of MMP19? Are there mechanisms that can cause activation to an active form of the enzyme, as there is for MMPs 2 and 9, for example, that activates the enzyme from an inactive form, in addition to just increasing or decreasing levels of expression?
There are a few studies with these enzymes, not many, but at least what we know now is that it is activated the same way as the other MMPs. Well, as the majority of the MMPs. That means that it's activated by proteases or some other molecules that get in contact with it.
Are there activity assays for MMP19?
No. And there are no specific inhibitors for MMP19. The problem with MMPs is that in general terms there are general activators, so it's difficult to just block one and to see what's happening.
Thank you.
Yeah, Annie, nice talk. I'm just wondering, given that MMP19 is associated with the increase in COX-2, does MMP19 cleave HGF from the membranes or from the extracellular matrix, or does it activate plasma? And I'm wondering if that could be the connection to the activation of COX-2.
We do not know, but it's a very good idea to say. Thank you.
Annie, that was a beautiful talk. Just in general, in addition to activating or inactivating chemokines or growth factors, have receptors analogous to the PAR system for thrombin been identified for the MMPs, where the MMP may clip the ligand, so to speak, off the receptor and directly activate a receptor? Is that been described for the MMP family?
I don't know in general. Probably for other MMPs there is something described. I don't know.
Okay.
For MMP19, no.
Sorry, I'm late again. Annie, I'm wondering, I may have missed the picture, but in your co-localization images of MMP19 and COX-2, were you able to see any MMP19 in the nucleus of the cells, or is it all cytoplasmic?
It's all cytoplasmic, but that's a very good question because some of the MMPs have been shown in nucleus.
Yeah.
And we looked for it, but